And here we go. Okay, good morning, everyone. I'd like to welcome you to the second edition of the virtual seminar series for the Centers for Iron and Heme Disorders at the University of Utah. And today we have the privilege of hearing from Dr. James Cox, who directs our Metabolomics core here at the University of Utah. James got his bachelor's degree at the University of Kentucky and then went to the Ohio State. How's that, James?
Yes, that's right.
Okay, where he got his Ph.D. in medicinal chemistry from in the laboratory of Nigel Priestley, where he studied bacterial antibiotic biosynthesis. And he then went to work at, was it a discovery company in Montana? I'm not actually sure of that.
Yeah. So we took my PhD project and turned it into a money-losing project or money-losing company.
Okay, awesome. And he joined the University of Utah as the director of the Metabolomics core in 2006. Since then has become the director of the Metabolomics and Proteomics core and is a faculty member in the Department of Biochemistry. And today he's going to talk to us about challenges in metabolomics of the erythrocyte. And with that, I'll turn the time over to James. Just a couple of housekeeping things. If you have questions, please put them in the chat and I will moderate that as we go along. And also, if you will all mute your microphones and turn off your videos, we will run better as the number of participants increases. Thank you so much. Okay, James.
Great, thanks, Ann. So all my talks, I really like to start off with the definition of what metabolomics is. Metabolomics is the study of all the low molecular weight chemical components of a cell, tissue, or biological fluid. And that sounds great. And it's a really interesting thing to do, but there's some real major caveats to this. And one of the major caveats is there's no single method that's fully capable of analyzing the metabolome. And so what we do to get a broad coverage of all the metabolites in a cell, we use multiple mass spectrometers, magnetic resonance systems. So we use gas chromatography, mass spectrometry, as well as liquid chromatography, mass spectrometry, and occasionally nuclear magnetic resonance.
James, I'm going to stop you for just a second. You've got quite a bit of feedback coming through. I don't know if you need to lean closer to a microphone or something, but there's some sort of...
Is this better?
It is better. Thank you.
And also flip the slides around so that we are seeing the speaker's view instead of the the view we should be seeing. Thank you.
Oh, thank you, Tom.
Right.
You see that view change?
Yeah. It's so hard. Okay, so what I do is do this. Take a second.
One more second and we'll get there.
All right, so PC screen only.
Bless someone.
Okay. Does that work?
We're still seeing your present
All right.
view. There we go. Perfect.
Okay.
Okay, thanks for saying that.
So no single method is, like I said, fully capable. And so lots of different mass spectrometry methods are used, basically. So another caveat is it's not necessarily sensitive. And what it's really measuring is the most high abundant metabolites normally in a cell or a system. And usually it's the micromolar range, millimolar range. We can get lower depending on how well metabolite ionizes. And one problem that we have currently is it's really not a rapid science. And the reason why it's not rapid is because we have to use all kinds of different mass spectrometry methods and extraction methods to get at the complete metabolome. And so what I set out to do is really to overcome these caveats, mainly because I got tired of projects lasting 3 months. And so the other what I really wanted to use was something that was simple, and that was the red blood cell as a model, because I thought, well, it's just a bag with a few metabolites in it. And so the other reasons why we did the erythrocyte is because we had a U54 grant in hematology. There's a number of researchers at the University of Utah who study various diseases of the erythrocyte. You know, Joe Burkall is on the call, John Phillips, Paul Segala. So it became pretty important to have a method to do this effectively. I was also approached by a sports doping agency to look at biomarkers of autologous blood doping. And really, one reason why is it's really easy to get red blood cells. But what I found was it's not so easy to analyze, which is fairly frustrating. And I'll go through some of those reasons why. But the major reason why is there's 270 million hemoglobin molecules per red blood cell. Basically, the red blood cell is just a bag of hemoglobin with a few metabolites thrown in there. And so what I wanted to do to make this faster is take inspiration from previous omics studies that have been done, other modalities. And this includes genomics, transcriptomics, and proteomics. And the reason why I really wanted to look at these is because they've been around a lot longer. So, you know, genomics has been around since the 70s, and Sanger won the Nobel Prize with a couple other people in 1990. But basically, you just have DNA or RNA in transcriptomics cases with 4 bases, and you can use PCR to amplify out signal out of those. And so that makes these 2 different kinds of omics, genomics and transcriptomics, fairly easy to get data out of low molecular weight or low amounts of sample. Now, proteomics is a little bit different. You can't amplify by PCR data out of or bases out of it, but what you can do is only have 20 amino acids, so it's a much more simpler compound than metabolomics. And what you use in proteomics is you just digest a trypsin, and you get a bunch of peptides. In this case, it's hemoglobin. I think you get about 40 peptides from hemoglobin, and all you really need to do is identify 1 or 2 unique peptides in proteomics to identify the identity of protein. So you can really use a lot less material in these 3 different omics because you can either amplify it out or it kind of comes pre-amplified. So looking at these previous omics and then looking at metabolomics and what the major reason why it's so much more time consuming to do is it really boils down to the chemical diversity found in the sample or in a biological sample. So just this series right here, these are just 3 carbon unit series. You can see propanoic acid, pyruvate, and alanine. They don't vary much. They just vary by 1 chemical component, but they vary somewhat by molecular weight, but a lot by pKa shown here and by the organic solubility shown below in the log P. You start adding some carbons onto this and you start getting a much more apolar compound or metabolite. And this is further complicated by metabolites that have different moieties added to them. So this is glucose, this is the glucose 6-phosphate, and this is ADP glucose. The problem with these in the mass spectrometer is when you run these, you'll see glucose, and you run glucose 6-phosphate, a lot of times these metabolites are labile or they'll fall apart in the source. So even though you're analyzing glucose 6-phosphate, this falls off in the source and all you observe is glucose. And the same thing goes for ADP glucose. And this happens to a lot of molecules. So you have a lot of complexity trying to interpret metabolomics based upon the fragmentation pattern. So what we have to do there is we have to run standards for every single metabolite that we can. And in our lab, I think we have like close to 2,000 metabolite standards that we can run to make sure we have the same retention time. This is further complicated by enantiomers and isomers, and the classic example of this is the D form of 2-hydroxyglutaric acid, which is found in glioblastoma, and it's classical, first named oncometabolite, versus the non-oncometabolite L2-hydroxyglutaric acid. So no metabolomics talk would be complete. I'm sure every single person here has seen a metabolomics talk, and every single one of them has this KEGG table here. I had to throw this on there just because I want to be like every other metabolomics talk, but also to illustrate all the different possible metabolites found in the human or in a sample. And so there's this great website, it's called the Human Metabolome Database. And what this group in Canada did is use the literature in their own lab and to determine how many different metabolites can be found in humans. And these metabolites include endogenous metabolites, drug metabolites, metabolites from the microbiome, pesticides, etc. And they have 142,215 entries to date. So basically what I'm getting across here, or trying to get across, is the molecular and chemical diversity that really makes metabolomics a challenge and really kind of slows down the process of analysis. And so in a typical analysis, just to illustrate this a little bit further, is these particular chemical components all have different pH labilities. In other words, glutathione, the reduced form, is stable at low pH, but is oxidized to the GSSG form at high pH. The opposite is true for NADH and NADPH. It's stable at high pH, but they are oxidized at low pH. Different components or compounds ionize differently at different polarities, ESI positive mode or ESI negative mode. So what happens here is if you want a complete analysis of the metabolome, we have to do 2 different extractions and sometimes 3 or more, and run 4 different LCMS analysis. A lot of times we'll do a GCMS analysis. And then if you really want to get at hard to get metabolites, we'll actually do solid phase extraction and get at like CoAs and things like that. So this has really become a burden. And so I'd rather ride my bike than sit in front of a computer analyzing data from multiple chronograms. And so what I decided was to do something about this. And this is just to illustrate the pH stability of the redox metabolome, which I'll go to here in a minute. As I said previously, glutathione's got a pretty good pH stability at 6.5, but it's completely falls apart or oxidizes at higher pH, and the opposite is true for NADPH. So as I was saying, I really wanted to get at reducing this complexity. And I really wanted to maximize the value of metabolomics, because if it takes 3 months to get your data, it's not of value. And so I'm concerned about metabolomics having value, and especially in my core, right? So the way about going about this is I really needed to collapse down the number of analyses that we do. And this is for not only to make things faster, but if you only have several methods, we don't have to have as many instruments we have. So we have like, I don't know, 13 different instruments because they all do something different. And so if we can really simplify the expense of instrumentation, this will save us a bunch of money, and we can pass that on to the user. Another thing, my second goal is really to increase the sensitivity of the analysis. As I mentioned previously, that it's not super sensitive in a lot of cases, and decrease the turnaround time. And as a scientific goal, and this is something that's bothered me for a long period of time, I really want to accurately measure redox metabolites in one analysis. And so in the past, we would have to do 2 different extractions, run 2 different mass spec assays to get at the redox metabolome. And if you look through the literature, a lot of bad data is out there for these redox metabolites because they're extracting all at 1 pH or analyzing all at 1 pH. And so what I started off to do, I started really set off to look at the hardest thing first, and that was the redox metabolome. And so what we did is we wanted to see how stable these metabolites really are, the redox metabolites, and confirm what we already knew from the literature. And so, but we also wanted to confirm what we want to know from the literature, or what we know from the literature, based upon mass spectrometry conditions, or how we do metabolomics. So the way we do metabolomics, we extract the sample, and then we keep it on the autosampler for anywhere from an hour to 36 hours, just depends on how long the run is. So they had to be, metabolites have to be stable as they sit on the autosampler, and also have to be stable during the HPLC process. So what we have here, what we did is we made stock solutions at NADPH, without NADP, NADH without NAD, and glutathione both oxidized and reduced, which I'll show you in a moment. And so we diluted these down into different pHs instead of on auto sampler, either put them at pH 9, shown here. This is the diluted pH as it sits on an auto sampler. 7, shown here. 5 here, and 3 here. And then we ran all these samples, all these stock solutions at different HPLC conditions. We ran them at 9, 7, 5, and 3. And what we found is that indeed the literature is correct, which isn't surprising. We found high levels NAD pH ratio over NADP if we keep it at 9 and run it at 9, shown here, or we maintain it in the auto sampler at 9 and run it at 7. And you start to see some oxidation, especially at the lower pHs down here. Same is true for NADH and NAD. We keep it at 9 and run it at 9, or keep it at 9 and run it at 7. Actually got really good results for NADH at the 7, actually really kind of tight. So the opposite is true for glutathione, which isn't surprising. The literature is true. Again, if we keep it at 3 and we run it at pH 7, we get really strong signal. So this doesn't mean that this guy right here is more stable. Actually, what we found was at pH 7, glutathione, the reduced form, ionizes super well. And so that's why it's higher than at 5 and 3. And also, it ionizes so well, we couldn't do the ratio of GSSG for here, so the GSSG over GSH, because the signal for GSH, even a minute amount, would distort these values, because glutathione, the reduced form, ionizes 100 times better than the oxidized form. So basically what we found, as long as we keep things in the oxidized form at 9 and run at 7, then things are pretty happy here. So we next did, and this is kind of hard to see, I just added this chart 10 minutes before I started this talk, is we then analyzed a bunch of different metabolites that you'd expect to see in the red blood cell, as well as other cells, and found here that in the positive ionization mode at pH 9, the best results for the most metabolites analyzed was found at pH 9, and that's this purple. And it's kind of a push at the negative mode, where you have majority of metabolites ionized better in the negative mode at neutral, but it's pretty close, 28 to 38. But one thing I want you to appreciate here is on this graph over here, is if we run things at the pH 9 through the LCMS, we get a vast majority of metabolites in the positive mode work really well. And then when we go to the negative mode, the converse is true. So where things are poorly ionized in the positive mode at 9 over here in the red, when we run in the negative mode, we get really good results. So you can see through this data that pH 7 and 9 in both the positive and negative mode are really optimal for analysis. And so what we decided to do at this period of time is choose pH 9 for the chromatography analysis and to maintain stability in the auto sampler vial. But really, what are we going to do with the free reduced glutathione? Because that's the fly in the ointment at this point, is that will oxidize. And so we decided to just derivatize it. And so this is pretty commonly used in the proteomics side of the world. So when we reduce and alkylate proteins to analyze by proteomics, we use iodocetamide or chloroacetamide to the sample, and it modifies or alkylates the free thiols in proteins. And so we tested these that we had in the lab, and also tested this ethyl propylate, which people are starting to use now to reduce or to alkylate thiols. In the past, I would alkylate glutathione with 2-vinyl-pyridine and analyze that by UV or by LCMS. And we also tried this N-ethyl-maleimide, which I'd actually never used before, that we found in the literature. And to explain this experiment a bit, what we did is we just took glutathione, the reduced form, put it in tubes, auto sampler vials, then added a 5-to-1 molar reagent of each one of these, iodocetamide, chlorocetamide. And then we put it through the mass spectrometer with each one of them. So what we found was, the idea was how long did it take for these to react completely? And we found that the NEM was completely reacted by 20 minutes. And the reason why we use this 20-minute point, because that's how long it takes to add the reagent, close the autosampler door, and then run a sample, right? And so we further analyzed it and found that NEM actually, under the conditions we use, under slightly basic conditions, reacted with free thiols instantaneously, which was great. So, and also a little bit disconcerting, like, so going back to the proteomic side, iodocetamide is not a great alkylating reagent. And so actually there's a paper out there I gotta go read about how to improve your alkylating conditions in proteomics. So that's great. And so the next step we had to do really to do quantitative redox protein or metabolomics was add some internal standards, right? And so this is the basic mechanism for glutathione, and this is an ethylmaleimide shown here. And so that's just a simple alkylation in this GSH NEM product. And so what we also do is we add a known amount of glutathione ethyl ester. The only difference is this guy here. And so this is completely non-natural. So we add that in a known amount. And so that will react at the same time with the NEM to form this guy. So we'll monitor this conversion from here to make sure that the reaction goes to completion. But it also has the added benefit is we monitor the ethyl ester GSH going from the reduced form that we purchase it in to the oxidized form. So now we can control for extraction conditions if there's any kind of weird oxidation that's happening while we're extracting the sample. And to monitor other metabolites, we use N15 labeled ATP. And what this does, we add this in at the same time as a control, and this will monitor for any kind of enzymatic activity that will convert ATP to ADP or AMP. So the goal here is to really have a measure of metabolic activity or to control for metabolic activity while we extract the sample. We also have in the sample, which I'm not showing here, a couple of different standards that aren't metabolically active, and we use those to control for extraction efficiency. So the next step was to apply all this to the erythrocyte. And so what we did, and this is just an aside, we originally were using red blood cells that we're getting from the clinic. So a clinician will often, in hemochromatosis patients, do phlebotomy, and we'll get the red blood cells as part of that project. Problem is going from the phlebotomist to the lab can take hours. And so we never observed ATP in those samples. And I knew it should be there, but it's pretty frustrating never to see ATP. And so what I did is I contracted with BioVT, which is a company that specializes in biofluids. And so what they did is they got human red blood cells and they processed the human blood cell as fast as they could, and then snap frozen and shipped it to me. And it cost me a bunch of money. But what we found we found actually ATP. So this goes back to sample handling is of the utmost importance. So taking these standard red blood cells that we have, we derivatized with glutathione by the addition of NEM and ethanol, and we found we can derivatize the glutathione as it thaws. So the next step is then we did a liquid-liquid extraction of that. And what we did is we add 450 microliters of 80-20 water to acetonitrile at 4 degrees Celsius with 0.1% ammonium hydroxide. Now this is a detail that's pretty important in metabolomics. It's completely opposite to how we do other extractions. So most extractions in our lab and almost everyone else's lab is the complete opposite, where it's 80-20 acetonitrile or methanol to aqueous. And the reason why that is you're trying to use the organic compounds to precipitate out the proteins to remove them from further analysis. But what we found were two things. One, even with 80-20 organic to aqueous, we still had hemoglobin that would clog up our columns. really possible to get rid of. A second aspect that we found, and we tested this in tissues as well as other biofluids, is the more organic you have, the less nicotinamide metabolites and glutathione, especially the oxidized glutathione, you extract. So really, to optimize our extraction efficiency for the nicotinamide metabolites and the oxidized glutathione, we had to go to a really highly aqueous solution. And so to prevent metabolism occurring at this really high aqueous solution, we incubate for 1 hour at minus 20 degrees Celsius. So we're hoping that cold will prevent metabolism and do some precipitation. We do get a small pellet after we incubate. But to remove the hemoglobin, the only really efficient way that we found, after trying many different ways, is we had to use an Amicon 3K filter. And so after much trial and error and many different filtration methods and precipitation methods, we had settled on this guy. And then this removes the hemoglobin. It's the only method we found that really gets rid of most of it, or almost all of it. And then we can go directly to LCMS. So there's a couple other advantages of using the NEM here. One, when you use the NEM, you're derivatizing the free reduced glutathione pretty instantaneously to get accurate measurement. But also, your NEM reacts with the free thiols of glutathione reductase and incapacitates that. So now we're preventing the oxidized form of glutathione being reduced down by glutathione reductase. So that's great. So we have a method for looking at the metabolome. Next, we look at the chromatography. We didn't really want to reinvent the wheel here. This is a method that we've been using for 6 or 7 years. We use sequent P-Hillic columns. The only difference is we use a larger size diameter column for the red blood cell, and we'll probably use this for other tissues, is there's so much glutathione in the red blood cell that it will saturate a smaller column. So this is why we use this bigger column, which is contrary to most metabolomics labs, which use smaller columns. And basically it separates by hydrophilic interaction liquid chromatography. So most metabolites are polar. Almost all metabolites are polar outside of lipids. And so you need something polar to separate something polar. So this column works by the sulfobutane column. So this is a really standard method used in metabolomics now. And the problem with that is there's such a high demand on this column, it's very hard to get. And so what we did this year is we just back ordered 15 columns and they all came. So we have a year's supply of columns, and it cost us like $13,000, I think, to get all those columns. So with that in mind, what the next step is we really needed to make the actual chemical analysis by mass spectrometry faster. And so just as a brief history of metabolomics, it really wasn't initially envisioned as a way to quantitate all the metabolites found in a cell or a tissue. It was really envisioned as a non-targeted discovery instrument. In other words, like you made a genetic knockout of a fruit fly and you wanted to see just what was changed and not anything else. And that made life fairly easy if you only want to look for biomarkers, because it's really easy to identify just a couple altered chemical compounds, right? If you're trying to identify 100 chemical compounds or 200, even if they're not changed in an assay, it becomes really, really laborious. So what I found was most PIs across the world don't really want to do biomarker discovery. As a matter of fact, I only have one or two, actually two projects that are doing biomarker discovery. Everything else is basic biochemistry. What are the levels of all my favorite metabolites in my cell system? So the QTOF became an unideal system, which is unfortunate because I have a bunch of these. Their advantages, like I said, they discover unanticipated and unknown metabolites. It's great for biomarker discovery. You can use accurate mass to identify metabolites with pretty high confidence. But there's real disadvantages. It's got a real high chemical noise. And this high noise leads to a low signal to noise ratio, which really leads to low sensitivity. And this noisy data sets really require high level expertise and a ton of time to work with. And that is a major problem in metabolomics. So the next idea I had, I'm not the only one doing this, is to do something called targeted discovery. And what it is we're using a triple quad instrument. The triple quad will only detect what you program it to detect. So in other words, it really won't do discovery at all unless you're doing a pretty broad assay for lots and lots of things. But because you're only looking at what you want to detect, it has a low chemical noise. And this low chemical noise really increases the sensitivity quite dramatically, actually, and you get a much broader dynamic range. The disadvantage is you only observe what it's programmed to scan for, and I'll argue a lot of times that's not a disadvantage. And it has traditionally a limited number of metabolites that are quantifiable at one time, but we have found that that's not necessarily true. Alan in my lab just did a lipidomics triple quad assay where he's able to quantitate like 640 individual lipid species. So we're getting into real high numbers of metabolites and lipids in a system. And so here's just an example of a QTOF chromatogram from the erythrocyte. And it may look fairly simple to look at, but underneath here, there's like hundreds and hundreds of different little peaks. And so the time consuming part of this is like, what's real and what's noise or what's chemical noise. And there's lots of different techniques that people use and we use to decipher through this. But the problem is it still takes time and a lot of time really to look through each one of these peaks and decipher what they are, right? So, and one other peak I want you to look at, this is glutathione, which I'll show you the same sample run on a triple quad here in a second. And keep in mind that this is pretty much equal in concentration across the board, this big peak. So in order to program that triple quad instrument, I went to the literature and originally my goal was to identify every metabolite that was found in the erythrocyte. And I figured it was going to take me, like weeks to do this because I'm really not a hematologist. I don't really know a lot about the erythrocyte, but I found this paper really quickly where that was all done for me for the 95% of all metabolites. In this paper by Bernie Paulson's group out of UC San Diego, we're doing flux analysis and modeling, really without mass spec. And they had gone through the literature and did all the work for me. And they listed in a nice Excel file everything that they found. And then I went and looked at Williams Hematology and here's Joe, people I know and wrote this part of this. But anyways, added some metabolites found in Williams Hematology book. So what happened here is I had found that, there's 400 or 500 metabolites between the two. Many of them we couldn't observe or didn't want to observe in this particular assay. Some of these were metals like calcium, etc. And so we don't do calcium analysis. That's something that's done by ICQMS, or lipids, which we actually have a completely separate platform for. So we end up with I think 400 potential metabolites found in a red blood cell that we could analyze by LCMS. And so what I did is I programmed in 135 of those metabolites into the positive mode, and we did this over a period of some time, and 104 in the negative mode. We tried all the metabolites in both modes, and this positive mode worked best for some, negative mode worked best for others. And we developed this assay, so the targeted triple quad quantitative assay. And one thing that you'll note here, even though it doesn't look like there's a lot of metabolites, there are a lot of metabolites here. One problem is the yellow doesn't show up well. But the point I want to make here with the triple quad is the glutathione is the largest peak by far. And when we compare that to the original QTOF, the QTOF was not giving proportional results compared to what we know the glutathione is. It's just a tremendous amount of glutathione here. So the triple quad has a wider dynamic range. And so you get better data out of it because it can go either higher in concentration or lower in concentration. And so I wanted to illustrate what this chromatogram looked like without the glutathione because it kind of distorts the the picture. And so I just removed the signal of glutathione, and what we see here, there's about 100 different metabolites, even though you can't see all of them, found in this chromatogram in the positive mode. Now, that's great and all. How does that help your research? Well, the best part about that is it's really easy. I mean, really easy to go from this chromatogram to a .csv file in a matter of minutes, whereas before it would take us, like 8 hours to do a data set. So in one data set now, can do anywhere from 5 minutes to an hour. It just depends on how complicated it is, versus 8 hours to a week. And so using this methodology, we're not going to find anything new metabolically wise, like we're not going to identify some new random metabolite, but in the red blood cell, there's probably not a lot of new stuff to be found. That doesn't mean that we can't go back to QTOF and run these same samples on the QTOF if you think there's something different, but this way the data analysis comes out way faster. So now that we have these handy dandy CSVs with our data, the next thing we really wanted to do is we have become saturated with samples and projects, and not just like, hey, we got a wild type and a mutant, here's 6 samples of each, can you tell me the difference? We're getting like sample sets with 1,000 samples, 2,000 samples, 3,000 samples, like 600 samples where we got 18 different conditions people want to compare, and it just became overwhelming to like, how do you deal with that much data, right? And so what we did is we used, I hired a postdoc through the U54 grant, Brad Naylor, who was a programmer. And so what we set out to do was to develop a program that would rapidly analyze these huge data sets. And so out there in the world of script and programs, there is a ton of different ways to analyze your data, but they're all written in like Python or R or MATLAB. And so you have to know, like 3 or 4 different computer languages and know how to use a command line on these to get any kind of useful data. And then you have to export that from one software package to the other. It just becomes unbearable. So I'll usually end up throwing something around in my office out of frustration because I can't get the right R script to work. So what I wanted to do was put all those scripts into a graphical user interface and call it something clever called a reporterator. And that's what Brad did. And so what we can do with this guy is we can go from that raw .csv data, and we've done this for like 1,200 samples at a time. And we can do a large number of different kinds of analysis through this. And we can take different files, we can take the adjunct files or science files or generic files that we have, and we can very quickly make a report. So it used to, once we got all the CSV data, like, and then it literally used to take us, 40 hours of work to like look at, a data set. And we're not talking about a large data set, like a data set of 1,000 would take us a month. But now with this, it does all the processing, the normalization, the chromatographic, aligning all the chromatograms, takes, a minute at the most, because we're using a really great software package. And so what that means to you, and it spits out a report. So it not only just spits out a Excel file for further analysis, it spits out a report. And so this report describes what you gave us, right, an introduction to Metabolomics, but more importantly, even though we don't have it here, you can cut and paste directly from the report into your publication. So the experimental. So the experimental should be absolutely correct. You can just take this, throw this into a publication, and then you don't have to ask me to write an experimental because it's already there. And if you want to analyze your own data, I got to point this out. There's freeware online. We based a lot of our software on this program called Metabolomics. It's freely available. So we go through the tutorial how to analyze your own data if you wish. And so what this report does is it spits out graphs like heat maps, PCA analysis, volcano plots. And it's super helpful for people to understand their data. And unfortunately, we don't have this yet. And I'm not really quite sure how we're going to do this, but it doesn't have a lot of pathway enrichment analysis. And the major reason why that is I haven't found a good script or a program that does pathway analysis for metabolomics well. And there's a reason for that as well. And that reason is there's just a lot of sparsity in metabolomics data. So even though like in the human metabolome of endogenous metabolites, there's probably about 2,000 endogenous metabolites, we're only observing, a 10th to 20% of the active metabolome at any one time. So, 70 to 80% we're just not observing. And so that creates a lot of holes in pathway analysis. So that goes back to, we need to detect more metabolites with sensitivity. So that brings us to this portion of it. So we're pretty close on publishing this. So we have to add and optimize a few additional metabolites. Like I said, we have like 240, I think. I'd like to do 300 total metabolites from the red blood cell. And the reason why, it just takes time at the order standards and run them by the mass spec. Do more in-depth comparison between the triple quad and QTOF. And we really need to validate this method. But the nice thing about this is we can expand this methodology to a lot of other biological matrices, the whole derivatization process. And so it's quite a bit different from the normal metabolomics extraction and analysis workflow. And I think we can get a lot of small publications out of it, which would be great. And so to conclude, we dropped down the method down to really one extraction method and two LC-MS methods, a positive and a negative mode. We increased the low abundance metabolite sensitivity, and we've really dramatically decreased turnaround time. And we can quantitate redox metabolites using really one extraction method and one analysis. So with that, I got a few extra minutes. I was going to show you something we're doing kind of similar in the proteomics arena, and then we can ask some questions. And so I gotta acknowledge my people. So Leon Ketro did all the most of the LCMS work, him and myself. The reason why this isn't quite finished yet is he had this little guy and was on FMLA. So people having children are killing my career. And so Brad Naylor did all the programming as well. And he actually had a kid as well. He's on FMLA. So Sandra Osborne does the proteomics, and Tyler does metabolomics with me, and Alan does all lipidomics. I'm sure many of you worked with. And so we have a number of funding. The most important one is this U54 from John. And so I have a few minutes, Dan. I can go through a little bit of proteomics, so I think it's kind of cool. You got a minute?
Yeah, let's do it. Unless, just want to pause for 1 second. If anybody has any questions that they want to ask, you're welcome to unmute yourself and ask them at this point. We'll just pause for like a minute, see if anybody has anything they want to ask orally. If not, we'll take 5 or 10 more minutes, James, with the proteomics information.
Hi, Diane. I have a question. This is Ella.
Wonderful. Thanks, Ella.
So how about species differences in doing this kind of metabolomics analysis? And the reason I'm asking this is a little bit out of left field. At some point, we were working with rhinos that seem in the zoo to be particularly sensitive to the kind of food they're eating and developing iron overload and having particularly fragile red blood cells. And I was wondering how easy it will be just to switch from one species to another if you have already optimized it for, let's say, human red blood cells.
Yeah, I don't think that'd be a problem. So the first thing we would do, we still do that QTOF assay. And what that is, that's just gonna identify all kinds of stuff that's in there. And so we would, try to compare what's different in the QTOF assay, and then take that data and port it over to the triple quad assay for speed. So we do a small discovery study to identify metabolites that are different in the rhino versus the human, and move those into the triple quad assay. Yeah, it would be pretty easy. And so because we can do that, I think we can do this for many different cell types. So my idea here is like cell cultures, one thing that's always annoyed me is every different cell culture's got a different metabolic profile. So it's kind of a pain to really optimize a lot of these things for each individual cell culture. So if we can have like a panel, like say male cells are completely different from, you know, like HEC293 or whatever. So have something very specific for male cells or CD34 cells, which actually are very metabolically dense.
Dan, I have a short question for James. James,
Sure.
great talk. I have reviewed recently a paper which is just published in Hematologica from Richard Van Wyk lab, which look at the pyruvate kinase deficiency metabolome.
Mhm.
And what they did is they took a fresh drop of the blood, 3 drops of blood on special filter paper, and got amazing amount of data using mass spectroscopy. I sent you that paper just in case you didn't see it.
Mhm.
But I was amazed how much information they got, and even getting really unstable substances, unstable metabolites on the really fresh sample, which now was preserved for weeks actually, and they have a very good data on that. Yeah.
Yeah, I haven't seen that. So my wife, believe it or not, does blood spot analysis. That's her whole gig. I would like to move to that. So that goes back to that looking at sports doping. First, I want to get like, you know, a good idea what's in the red blood cell, what we can see, and then move that to the paper. But you know, I have to look at the data to make a real comment on it. I think, yeah, I'd have to look at that data. That is something that people are doing, and like I said, my wife's doing that as well. But it would be really convenient because there's just tons of blood spots out there that people can go back and look at, especially if it preserves well.
Yeah, I just sent it to you. It's from very reputable lab actually.
Cool.
Any other
Yeah,
questions?
I'll just make a comment. The reason why we're using 50 microliters is because that's our filter works best at 50 microliters. We could always use less packed red blood cells. We could probably use a 10 or less.
James, just as a clarification for those people who are watching to understand preps and sampling, do you have any suggestions for that? They provide you with frozen what?
So the best way to go about any of this is to, you know, talk to us first. But the most important aspect of sampling is freezing metabolism at the point of sampling as soon as you can, and second most important thing is doing everything the same way every day. So consistency is the most important thing actually after freezing metabolism.
So then you would recommend people contact you as they're planning forward for these kinds of analyses.
Yeah, because like liver tissue and things like that, we really try to take, you know, if you're doing liver, you really want to take specific lobes or the same lobe every time. Unfortunately, a lot of times, like people have a bunch of liver stored in their sample from a project, and they're like, hey, let's go look at the liver from metabolites, but it's very hard to like chop off the same part of the liver lobe once it's frozen and stuffed in a vial. So thinking ahead a little bit is, if you're going to do metabolomics, like, you know, section out tissue and then freeze them individually, like individual lobes. Sometimes that's not possible.
Okay, any other technical questions there? We're kind of at 50 minutes, so maybe we will pass on the proteomic piece and give you an opportunity to tell us that on another time. Any other questions in the chat or just orally? You can unmute yourself and ask a question. Otherwise, we'll end the presentation. Okay, I think that is it. James, thank you again for the presentation. And we look forward to seeing everybody again for our next talk, which will be given by Ellen Namath. Everybody take care and stay safe. Thank you again, James.
Thanks.